like to welcome our next speaker, who is our very own research scientist, faculty in our Center for Agroforestry in the School of Natural Resources. He's actually quite a powerhouse, very prolific scientist producing amazing work in the area of bioprospecting, bioremediation, and also work with useful or extraction of useful phytochemicals from plant materials, especially working with some of the specialty crop native fruit and nut trees and shrub specialty crops that our center works with. So please welcome Dr. Chung-Ho Lin. Looks like they're still setting up the mic, so just 1 second, please.
Hi, thanks everyone for coming to my talk. I really appreciate it. So I'm going to bring you some of the highlight from our center research project and keep you updated how we got involved into this translational research. Okay. Before we talk about the research, I would like to use an analogy, how to find a new idea. So my analogy is how we can find the new species. I use that same analogy for my project. But keep in mind, this is just my own opinion. No, that might not apply to everybody's. So how we can find a new species in the ecosystems? So this is a highly managed system. This system was designed at the beginning with one single goal: to maximize the productivity of the commodity crop. Under such high selection pressure, occasionally you might find a species such as a new wheat that are resistant to every herbicide. So this system is not sustainable. When the selection pressure is gone, you lost any chance to find a new species. So I won't try to find my new species under this system. Which system I should go? This is system. So why? Why I think that's the best place to find the new species? I will explain. Because this system offers a huge diversity, also is self-sustainable. So to make this system work, you need to have a driving force like evolution, right? What kind of driving force to generate a new species? Actually, I can come into this system to find a new species every day, okay? Those are the driving force for this evolution process. Desire, could be greed, can be desperation, can be competition, can be symbiotic coevolution. That's one process I admire the most. I scratch your back, you scratch my back. And we work together, we co-evolve, and we generate a completely new species. Also, sometimes you need luck, okay? Such as a natural disaster. It can be fire, it can be drought, it can be flood. Therefore, under this system, it's self-sustainable. And in this system, any crisis can be the opportunities. Okay, sometimes because of the fire, right, they help you to generate the new species. They can tolerate the heat, tolerate the drought. So that's the model I've been using for my research project. So I would like to talk about diversity. By training, I'm more like analytical chemistry. So I consider the MU campus as a rainforest, okay? So I try to find everybody to scratch their back. Hopefully one day they will scratch mine as well. So I try to overlay the different disciplines on campus, including the bioinformatics, molecular biology, material science. And I only dive into this place where they overlap. That's the new species for me. So I only pay attention to this area where the discipline overlap. Okay, I'm going using this approach. So far, there are 3 spin-offs from this collaborative platform. One is the environmental enzyme. There was found by Brian, Katie Thompson. I'm not going to. They're going to be the next speaker, so I won't touch anything. Okay, I won't. And another one is the Tiger Energy, focused on the production of a cellulosic bioenergy. Another project they call Black Diamond. It's a spin-off from a student project. She spent 2 years without program. She decided to take what she discovered from her master's degree, turning into a business. She's in the early stage, early process of trying to license that technique from the MU Technology Transfer Office. And she start up this company, and right now she joined a Korea pharmaceutical company called Prime Pharma pharmaceutical company, and continue this line of research project. Why it called Black Diamond? Any idea? Guess. Good guess. Okay, she was working on the coffee, spent ground coffee, how to turn the spent coffee ground into some product that can be used in the cosmetic industry. Black, that's the color of coffee. Diamond represents beauty. So I'm going to take the coffee as an example because it is early morning. Let's start with the coffee, right? You're drinking coffee, you're testing coffee, you're going to listen to the presentation associated with coffee. Hopefully that will help you, keep you awake, okay? So coffee, there are more than 6 million tons per year of spent coffee ground generated worldwide, okay? Right now, the only use is for compost, right? People all go to the trash, right? People consider that as waste material. But from the perspective of Mother Nature, there's no such thing called waste material, right? If you put it in the right time, right place, they could be highly valuable products. Depends on how you assemble the approach and idea. So 2 years ago, we were trying to find the new use of this we call waste material. But if we decide to use a traditional approach, take probably 3 generations. Because if we use a traditional bioassay guide purification, probably within 2 years or 3 years, you can only identify probably 10 compounds. Probably you can identify less than probably 15 applications. I won't have that much of a time. So we decided to utilize the new approach. Fortunately, 3 years ago, L'Oreal started bring his expertise to the MU campus, established a metabolomic core facility. So I feel I was fortunate. Okay, so we start with the high throughput screening process. What does it mean high throughput? Basically, they use a robot to do the bioassay. Right now, they have a capacity to process 300,000 bioassay per week. So when you're sleeping, they're doing the experiment for you. Every bioassay only takes less than 10 nanoliter. So basically, there's only one bubble they can do the bioassay. So they use this, we're using this facility to identify every possible application for this waste material. At the same time, we inject our extract into the Loy's core facility. One injection, 15 minutes injection, generate 5 gigabyte, 5 to 6 gigabyte of high resolution information. That they are the high resolution chemical fingerprint. Then we upload and submit those fingerprints to the Scripps Research Institute in San Diego. They have the platform using the server equipped with 1,400 CPU to process that data. So you can check your data on your cell phone, on your laptop, on your tablet. It only takes about 3 hours. They can help us identify more than 250,000 of chemicals. Then our students spend the time to go through this data to identify those compounds might have a commercial application. So this can give you some idea. We submit a fingerprint we acquired from Lois Lab, submit to this XCMS platform. They will do all the deconvolution, they will pick alignment, pick annotation, and compare that fingerprint with a fingerprint in this Malingerer Library. This Malingerer Library is one of the most comprehensive library for natural product in the world. It's free. It's free. Everybody can access that library. So it consists of every natural product has been identified in the human history. Those products originally isolated from plant, bacteria, fungi, mice, human. Okay? So with that process, we can easily identify 200 to 500 bioactive compounds from this waste material. Then in my lab, we did a LC-MS analysis to confirm and quantify this chemical. Yeah, we have poster in the hallway. You can take a look. If you're interested, you can stop by and take a look. Okay, we're using the same approach for other plant, they call it waste material. Okay, I'm a big fan of turning this trash into the cash. I borrowed that statement from our centerdirect.go. So if we can turn this waste material to some high-value compound, it immediately generates the revenue for the producer, right? We're particularly interested in personal care products. Why? 5% of the active ingredient you can put a shelf for at least $50, and 95% of that is water. Actually, you are selling water for $20 per bottle, right? Dietary supplement, you're charging probably $6 for probably 50 grams in a bottle. We think that's an excellent model. At the same time, they're organic. Actually, it's win-win. The landowner, the producer, they are happy, right? As a researcher, we feel rewarding to see some of the product into the market. More importantly, they make every woman happy. When women is happy, their husband is happy. So I think we are making a huge contribution to the society, right? So using that approach, we have identified several, don't laugh. we identified several chemicals that have been formulated into the personal care product in the industry. And also there's the student who started this Black Diamond company, okay? She also learned how to do the formulation in our lab, then try to turn some of their benchtop discovery into the market, okay? During that process, we also learned a lot of compounds. They are not active because they are because of this attachment of glucose. They are in a glycosylate form. So actually, they are the activity, anti-inflammatory is strong, but because of this sugar attached, we never be able to release their true power. We learn, using some enzymatic process, we can cut off this sugar and turn that into the bioactive form. Here's another example. Some of the compounds, they are active, but they cannot be easily formulated into the formulation. And also, sometimes they are having the problem with that stability. Using the enzymatic process, we can convert that into the ester form. Ester form is more active, more stable, and can be easily formulated into the lotion. More importantly, you are turning into some low value, low active ingredient into the high value product by a single enzymatic process. So that led us to think about whether we can develop like an affordable enzymatic bioreactor system to catalyze this reaction. But traditionally, this is cost prohibitive because one is the enzyme purification process is so expensive and labor intensive. Also, the chemical conjugation of enzyme to immobilize the enzyme under any solid surface, that's also cost prohibitive. So by working with Dr. Xie and Dr. Stewart at Life Science Center, we try to we develop one approach. Basically, they developed the genetic cassette. Okay, we can express any enzyme that we're interested. This is the example, their product. Every enzyme will be genetically fused with a protein linker. Traditionally, we have to do that manually, but right now we rely on the E. coli to do all the job. This is the product. The enzyme, every enzyme, they will link with a linker automatically without any effort. So that eliminates almost 90% of the cost. I will explain later, okay? Then we create a functionalized platform, okay? Because this specific interaction between the linker and the functional group and platform, we can easily immobilize it, okay? That completely eliminate any cost of enzyme purification. Also eliminate any cost associated with chemical conjugation. The beauty of this system, when the enzymes start to denature, we can regenerate this service. Keep using the same platform over and over again. So imagine this is like a cartridge. We can keep loading the enzyme. Now, use that as delivery system or bioreactor system. Then after the enzyme start losing their ability, we just load another fresh of a recombinant enzyme. So our team has tried successfully using this approach to immobilize the enzyme and service the carbon fiber, polypropylene, polystyrene, nylon, glass fiber, silicon, carbon nanotube, even magnetic particles. So what's the major advantage of a system? Because that specificity of the interaction, we eliminate the cost of the expensive time-consuming purification process and chemical conjugation process. Another advantage on the surface, all the enzymes are pure. There's no impurity. Every enzyme is pure. And the system can be reused and regenerate. We can immobilize a single and multiple class of enzyme. Also, the platform will help to enhance the stability. Also, the whole system is programmable. My generation, when I learn about programming, we learn the C++, basics. So this 0101 binary code. Nowadays, when you look at the programming, actually you're talking about programming with ATCG. So this whole system is programmable. We can express any enzyme that we're interested with that linker immobilized on the surface. So that's one of the first prototype we use. We call this active fiber. This is a carbon fiber coated with all every enzyme we immobilize, they are pure. So we can use this fiber as the delivery system itself, or we can pack it into the column, make it into the continuous flow reactor. So that's one of the example. So basically, we pack in the column, we just circulating the substrate, and all the substrate are converted into the product if you see the color. Okay. Also, we immobilize that on the magnetic particle. Then using magnetic block, we can recover it. So this is enzymatic activity. And at the same time, we try to, one of the student, Mason, he's helping us to develop a platform using the 3D printing techniques. So there's some of the prototype. The idea is we want to create a cartridge, just like cartridge for your printer, right? Every household, they can take that cartridge, probably dip in the solution. They can probably do that at their home. Okay, and this is the first generation, second generation. The idea is that we want to create a platform with enough surface area to allow us to immobilize that enzyme and create the enzymatic bioreactor. So this is the 15th generation. So hypothetically, using the 3D printing, we can construct a platform in any size, any form, any shape, fully customized. Imagine, now we're thinking about the wound healing mask. Because a lot of enzymes, they can serving as a therapeutic purpose. So we basically can print the mask, exactly match the shape and curve of your face. That's our ultimate goal, okay? So this is the one of the final prototype. Now we create this cartridge. They give it enough surface area. We immobilize the enzyme, drop into a column. We circulate the substrate. And here, then we can see we keep producing the product. This is the concentration of the product over time. So basically, all you have to do is just keep circulating your substrate. Everything will be converted to the product. Why we put so much effort at special chemical? We strongly believe once we can prove our concept on special chemical, they'll put us in a very good position to move into the pharmaceutical industry. Right now, there are 500 drugs in the market that require enzymatic process. Why? The industry realized using an enzymatic process, they got a better yield. Also, it eliminates a lot of toxic chemical in the biosynthesis, the synthetic process. So I think once we have a prototype delivered, we can position ourselves to take our prototype, extrapolate that into the pharmaceutical market. There are other applications, okay? This work originally was initiated by the Professor Dan Smith and Dr. Shea at the MU campus, okay? People have different blood types. One of the reasons that make a different blood types is because there's sugar, the antigen, on the surface of red blood cell. Okay, for example, if you got a blood type A, you have a blood type A antigen. This is sugar. If you have a blood type B, you have blood type B sugar. And for O, basically, you do not have those antigens on the surface. Then Dr. Shen And Dan Smith, they identified several enzymes from the bacteria, Clostridium. They can cut off this antigen on the surface of red blood cell. So we got that gene sequence expressed with our system with linker, immobilized on our bioreactor. Now our effort is to cut off this antigen, make all the blood type A and blood type B into the universal blood. Okay. So this is our first prototype. We immobilize that system. We start to see the conversion. So most of the blood type A got converted to the universal blood, O. At the same time, as what we have learned, red blood cells, they are fragile. They are vulnerable to the mechanical stress. So we use the fluid simulation model to optimize our system to minimize any shear stress of red blood cell in the system. So this is the result of a simulation. So in this system, we were able to maintain the integrity of red blood cell during that conversion process. Another application, you remember we were talking about the bioactive fiber, right? They can be excellent carrier for bioremediation purpose. So our lab has been working on the several enzymes like ATZA, TRZN. They can break down the atrazine into the non-toxic degradation product in one single enzymatic process. Why we care about atrazine? We're living in the area, the Corn Belt. Every year they apply 500 million pounds of atrazine in this area. So during the early April to August, you can find the atrazine in the rain, in the water, everywhere. Okay, so we're thinking, and almost all the drinking water well, you can find the atrazine in there. Why we're concerned about atrazine? Atrazine behaves like an endocrine disruptor. Because of that, it has been banned in Europe except Spain. So with this bioactive fiber, if we can pack into this mesh container, drop it into every contaminant well, because of that electrostatic of the carbon, they will attract those pollutants. And the enzyme on the surface will help to break down those atrazine. They can be used to clean up every contaminant well in this Midwest region. So we did submitted several SBR proposals, try to propose that approach to the USDA. And when I started my academia, I thought this was my war, publish or perished. But I realized, if I'm lucky, by the end of my career, I might get like 200 publications. If I'm lucky, I might get 200 citations for each of my publications. But I think it still takes a long time to see the impact. So I decided to go to the jungle, okay? Find my partner to extract some idea. And I was hoping with this idea, I can turn it into something that can benefit millions immediately. So this is my version. By putting the right brain together, right team together in this jungle, we should be able to develop a strategy to change the way we produce the food, change the way we produce the medicine, change the way we clean up the environment, change the way we produce medicine. Fortunately, with the help from Brian and Katie, we accomplished one of the goals already. So I would like to acknowledge all my collaborators in this rainforest, okay? Don't forget, next time if you want to find a new idea, go to the jungle.
Thank you so much, Chung-Ho. That was a great presentation. We have some time for some questions, so start right here, Dr. Sumner.
Very interesting presentation. I appreciate that. The bioreactor with immobilized enzymes, that reaction can be done chemically as well, just through acid or base hydrolysis.
Yes.
So have you looked at the cost of doing it chemically versus enzymatically, and is there a benefit of one over the other?
Yes, I did. For example, for remediation, there's some redox oxidation process. They can also get rid of some of the pollutants. But at this point, at early stage, we target some area we think we can bring some solution more like an environmental friendly approach. Okay, and also our ultimate goal, we really want to get into the market where the product with high value, they can justify the cost of our manufacturing process.
I think we have one question back. Am I seeing a question? Well, yeah, you're on now, Katie.
A little bit, but it's the, in our case, it's the specificity of the reaction,
Yeah.
which is certainly important because in our case of the soil, where our reactions are occurring, you can add acid or base to the soil, but you're probably not going to get the outcome that you want to achieve. And so in pharma, you know, Chung-Ho can probably speak more to the specificity there, but enzymes, they definitely provide a more sustainable, more specific reaction that's going to produce less caustic byproducts.
Nice to have feedback from industry.
Okay, thank you, Katie, for that observation. Looking around, we have perhaps time for one more question. Is there anything for Dr. Lin? If not, then thank you for your staying with us, for your participation, and thank you to Dr. Lin for that excellent presentation.